Thank you very much for that lovely introduction. I'm based in Western Australia and I'm part of the Walian Respiratory Research Centre of Excellence. Walian Walangup is an Indigenous term for place of healthy lungs, and we're a global epicentre for paediatric research. And today I'll be talking about a project looking at prenatal risk factors for lung development and how we might conduct epigenomic studies to understand how early life events really set the scene for future risk for respiratory morbidity. I'd just like to start with this analogy. These two boys here are my nephews, Sammy and Raf. They're about 2 years of age apart. They share about 50% of their genome, like all siblings do. And they grew up in the same household. They eat similar food, and during winter, they experience the same viral illnesses. But Sammy's respiratory health is different to his brothers. When he gets a cold, it triggers wheezing episodes, which sometimes lead to hospitalization. He is vulnerable to viral inflammation, although he's too young to actually receive a formal diagnosis of asthma. He exhibits many of the features of a child on that trajectory. And unfortunately, there really is nothing we can do about it right now. There's no practical advice that we can give to his parents. We're stuck in this scenario of wait and see if it develops into asthma before treatment is initiated. And unfortunately, Sammy's not alone. Respiratory diseases are still one of the major causes of disease morbidity globally. These are burden of disease statistics globally around the world. And among the pediatric population, it's one of the most frequent causes of hospitalizations. And so while we've had really substantial gains in asthma treatment, you can see from Sammy's story, we need to shift the focus towards disease prevention. And so that's one of the key areas we're working on, is around biomarkers for early identification of respiratory vulnerability and developing novel treatments that can prevent asthma from developing in vulnerable children like Sammy. So epithelial vulnerability in children with asthma, our colleagues in the Walian Centre have done a lot of fantastic work characterising respiratory vulnerability using nasal epithelial cell cultures from asthmatic and healthy children. What I'm showing you here is data from nasal epithelial scratch and wound repair assays showing that children with asthma have intrinsic deficits in their respiratory epithelium cellular repair and migration capacity. So the team have been really successful in showing that there is a cellular phenotype here which can be studied in the lab using respiratory nasal brushing cells that recapitulates intrinsic vulnerabilities of the airways. And what we want to understand is the nature and the origin of this vulnerable and epithelial endotype. When does it begin and what are the mechanisms? Unfortunately, the state of science is that we only have a really high level of understanding of how airway epithelial vulnerability arises and why it happens only in some children. What we know is that it involves complex interactions between the genome and environmental risk factors that happen during very sensitive periods of early development. And in fact, several of the asthma GWAS studies have identified that the risk variants are related to epithelial biology. So at a very basic level, epithelial vulnerability seems to arise from gene-environment interactions, possibly mediated through epigenetic mechanisms. So the aerial birth cohort, which I'd like to tell you about now, was launched by us in 2018 to try and answer some of the key questions including whether the vulnerable epithelium is programmed during fetal life and whether we can leverage molecular profiling studies to identify new biomarkers and targets for intervention. So the aerial birth cohort is nested within the Origins project, which is a birth cohort of 10,000 families recruited during pregnancy and followed up until their offspring are 5 years of age. So for the aerial study, we recruited 300 mother-infant pairs and we collected both nasal respiratory samples and placental samples, matched placental samples from each of the newborns. And we're monitoring viral exposures over the first year of life, including the nasal microbiota, and characterizing pathogens. During those infections, we're recording symptomatic illness. And beyond 1 year of age, the children will be assessed for respiratory and allergic phenotypes, and we're hoping to extend the cohort out to school age to measure lung function. So what Aerial will allow us to do is to identify genes and pathways involved in the fetal programming of disease risk and follow those markers up over time to see how they're modified by viral and other exposures, and how they relate to disease inception, with the ultimate view that we can hopefully use these markers to identify high-risk kids early and recommend strategies to build resilience in the respiratory epithelial tissue before it's too late. So the data I'd like to show you today pertains to a very specific question about whether the placental amnion tissue that we're collecting is useful for studying how gestational exposures can affect respiratory epithelium development. So we've been working with the nasal respiratory tissue, as I mentioned, to characterize respiratory vulnerability for some time now, but it's not always easy to collect this tissue from newborns. We often need to wait several weeks after birth to collect it, and by which time the epigenome is continually modified by the colonizing microbiota and the environment. Now, the amniotic membrane is a fetal tissue, which you can see here, that is exposed to the gestational environment, and it's rich in epithelial stem cells. They're known as amniotic epithelial cells that are recoverable from the lining of the inner membrane of the placenta. And these have got stem cell-like properties. So these cells can potentially provide a window into gestational events that impact epithelial and stem cell development. And one of the key things we're interested in is whether we can identify signatures of disease risk that can be followed up in the mature respiratory tissues as children age. So we designed this proof of concept study within Aerial. We harvested amniotic epithelial samples from 88 newborns and matched nasal respiratory samples that were collected between 2 and 6 weeks of life. So every newborn had a matched pair of samples. In total, we had 176 samples that were collected, and we extracted DNA and RNA from the tissues, which we've used for methylation sequencing and transcriptomic analysis. Today, I'm only presenting the data on DNA methylation, but our laboratory lead, Nina Krasoye, who's an exceptional pair of hands with NGS in the laboratory, built 176 libraries, which were sequenced on the NovaSeq 6000 at 2x150 base pair on the S4 flow cell using an XP workflow. So in total, we generated at least 160 million reads or sequences per sample through our genomics research partner, Genomics WA. So we typically use microarrays for our large cohort DNA methylation studies, but we chose to use the Twist human methylome panel for a few reasons for this study. One, it doesn't involve bisulfite conversion. It uses an enzymatic conversion. And that actually meant that we could get away with a lot lower DNA yields. And that was important because we don't recover a lot of cells from the amniotic tissue, and most of it needed to go into the transcriptomic assay. Secondly, because of the nature of microarray data, which is fluorescent intensity, we wanted the quantitative sequencing reads because of the greater sensitivity around the extremes of the bimodal distribution that you get with sequencing data, which gives us a greater sensitivity, given that we knew we were looking for small effects. And finally, the additional content of the Twist panel substantially expanded our search space, allowing the flexibility to scale NGS workflows up to several hundred samples, at a similar cost without being completely buried in the data as if we would have been if we'd gone whole genome. So developing the bioinformatic capacity did take some effort because we hadn't worked with this particular data before. We processed the FAST-Q files using the NF-core MethylSeq standardized bioinformatic pipeline that we've modified to include a Picard profiler tool. And that allowed us to collect metrics on the alignment files around coverage and uniformity of the sequencing assay, and it's conveniently compiled into a multi-QC report. And the process data were analyzed in RStudio using the Bioscience bioinformatics package. And the bioinformatics pipeline, we've implemented this modified version of the pipeline in the Nextflow language, and we've made the repo, the repository, publicly available to anyone who wants to use it. And we're happy to share our experience with that if anybody is interested in using that and reaching out. So the bioinformatic pipeline was implemented on a high-performance compute environment at the Pawsey Supercomputing Facility in Western Australia. All of this was tested and developed by Patricia, our bioinformatician, and we received a lot of fantastic support from Ananth and Adam and the team at Twist to help orientate our compass as we worked through the bioinformatics with this new data type. Okay, so looking at the Picard metrics, we had good coverage and uniformity. This is the fold 80 base penalty, which was between 1.2 and sort of 1.6, which for these low diversity libraries is good. The average sequencing statistics, when we compared the nasal samples and the amniotic samples, was similar, with more than 90% of the bases that we'd sequenced were covered by at least 30x. And here's just an example of the alignment files that we get from two randomly selected samples. This is the nasal and the amnion tissue across a gene here. So you can see we get quite a lot of coverage across these regions. The methylated reads are shown in red and the unmethylated reads are shown in blue. And you can see a good example here of a region where the levels of methylation are different across the different tissues. So looking now at the general features of the methylation landscape, we could see that tissue was the major source of variation in this data set, with the amnion samples clustering differently to the nasal respiratory samples. This is a PCA plot around gene promoters. What we can see here is the amnion clustered quite tightly, whereas quite a lot more variability in spread in the nasal respiratory tissue, which likely reflects the effects of the colonizing microbiota and environmental effects as well, given these samples were collected, sort of up to 6 weeks of life post-birth. Despite these differences, there are still a lot of similarities. This is the sort of distribution of reads around the transcriptional start site, distribution of methylated reads around the transcriptional start site, which was pretty similar between nasal and amnion tissue. And when we looked at the CPG islands, we can see that CPG islands were similar between both tissues, where we saw patterns of fully unmethylated and methylated islands in similar frequency across the tissues. So using differential analysis, we identified that there were about 4,500 differentially methylated regions, or DMRs, between the tissues, which is depicted here in the volcano plot. Each point on the plot here is a DMR with the size of the plot shown here. So we have a range of sizes of DMRs. And on the x-axis, we can see the effect size of loss or gain of methylation. And we determined that about 20% of the genome was significantly different between the tissues. You can see an example of two of these DMR plots here, where we see, for example, this is the level of DNA methylation in the nasal tissue. It's fully methylated, whereas in the amnion sample, we have low levels of methylation across the gene body of this particular gene here. These are the individual replicates. So we can see here the difference in the methylation patterns that we get for a few of these DMRs that we're identifying. And so what we can do with this data is we can remove all of these tissue-specific regions in the genome, and what you're left with is the genomic regions where the methylation patterns are highly concordant between the tissues. And so now we can limit our search space to within these regions, which is sort of the remaining 80% of the genome. And within this highly conserved fraction, you can see the correlation of methylation levels between amnion and nasal are highly correlated within this fraction of the genome. Within this, we identified 159 genes that are uniquely expressed in lung tissue that are shown here in the heat map. So these are log2 transcript counts from 32 tissues taken from the GTEx consortium database. So for this set of genes, which you can see is very unique to the lung, or they're very unique to being involved in lung development, the methylation levels are highly concordant in both tissues. So here's an example of two of them here. This is a surfactant protein gene, which demonstrates intermediate levels of methylation. And here are three of the nasal and amnion replicates that show very similar methylation profiles over this region. And this gene down at the bottom here, SCGB3A1, is an epithelial-specific gene regulating cell proliferation, and this is largely unmethylated in both tissues. So what this means in practical terms is that the epithelial cells from the amnion can be used to study how gestational exposures might disrupt DNA methylation at genes relevant to lung and epithelial development. And so as a proof of concept study, we looked at two very well characterized gestational exposures that the epidemiology literature tells us increase the risk of developing asthma in the offspring. The first is maternal smoking, which we measured from urinary cotinine samples collected at 20 weeks of gestation. We only had 4 individuals that of the 88 that were smokers. But we found that there was a signature of 111 differentially methylated CPGs that were clearly established very early in development, because the methylation profile of these markers is very different, both in the amnion and the nasal tissue. So this must have been a programming event that has occurred very, very early in development. We also looked at maternal asthma, which is again a well-described risk factor for offspring asthma risk. Here we had 9 mothers with asthma, so slightly more, and we found a signature of 17 differentially methylated CPGs that were again imprinted on both the amnion and the nasal tissue within that conserved part of the epigenome. So these sample sizes here for these exposures are small, so it's really encouraging that we can see these effects, and likely we would see more associations with a larger sample size, but it means that we can confidently say these programming events occurred in gestation, not postnatal life, and we can now track these signatures through time in the nasal respiratory samples as children get older to see how the early environment modifies these signatures and how they relate to the eventual development of respiratory disease. And finally, we mapped these individual signatures to genes, and the enrichment testing showed us that from the maternal smoking CPGs, they were enriched with human phenotypes related to increased inflammatory responses and abnormality of the immune system, which is a feature of what we see in children who develop asthma, as well as terms related to gastrointestinal epithelial development and viral responses. And for maternal asthma, very interestingly, we saw enrichment of genes involved in wound repair, which very much recapitulates what we see in the epithelial cells. We also see genes involved in lung morphogenesis, and tissue migration pathways, very much recapitulating what we see in the cellular phenotype of these children. So I'd just like to make a few acknowledgements here for my research team, in particular Nina Crusoye, who did all the NGS work for us in the lab, and the aerial investigator team. In particular, I'd like to mention Professor Stephen Stick, who really has led the way with his team in conceptualizing the vulnerable epithelial hypothesis, and was instrumental in setting up the aerial birth cohort, but also Liz Starcevich, who helped with collecting the samples and managing the program, Patricia, who did the bioinformatics, as well as Thomas, who led the way in the scratch wound repair assays, and the other investigators. I'd like to acknowledge our research funder, National Health and Medical Research Institute in Australia, and our research partners at Twist Bioscience and Genomics WA. So just to summarise, hopefully I've shown you that the amniotic tissue is a promising surrogate for the nasal epithelium. It showed a high degree of concordance in the non-tissue specific regions and may be a good accessible tissue for studying prenatal programming. This conserved methylome region could hold the key for understanding how early gestational exposures influence lung development, and how they influence amniotic and epithelial stem cells as a starting point for understanding how respiratory disease inception occurs. And finally, our proof of concept study may support the idea that there's a possibility for biomarkers within the placental fraction for monitoring future disease risk. So thanks very much for your attention, and I will end the screen share there.